Welcome back from lunch. We're about to start the second part of our Biobanks for Precision Medicine. We had a great, I feel, and I'm biased, a great session this morning. This afternoon, we're going to be learning more about biobanks in Asia, predominantly, and particularly focusing on their ability both to drive mechanistic discovery research, but also to translate that research to clinical applications. I'm going to be brief with bios. I really want to give our speakers the most time possible on the podium, but we're going to try to keep things on time this afternoon. So our first speaker is Professor Dr. John Chambers. He's Professor of Cardiovascular Epidemiology, the President's Chair at Nanyang Technical University, LKC Medicine, and he's the Chief Scientific Officer for the National Precision Medicine Program, otherwise known as PRECISE. John?
Okay, the clock is ticking. So my name is John Chambers. I'm the Chief Scientist for PRECISE. That's what I do in terms of helping coordinate programs. But I'm also an epidemiologist. That's what drives me. And what I'm going to talk about now is how the motivation to solve problems has driven us to create a cohort, and then show how we've used that cohort to solve the problem, what from my point of view is one of the key global problems. Okay, so it's functional genomic insights in Asia, design and exemplars from the PRECISE S200K program. Okay, so as an epidemiologist, a non-communicable disease epidemiologist, I'm interested in diseases like diabetes, cardiovascular disease, which are well known to be complex and multifactorial. And the thing about complex multifactorial diseases is they are complex and have multiple causes. It's not just genetic, it's also behavioral, it's also environment, it's a range of other exposures. And whilst the genome is fundamental to, on average, 30% of our health or health variation, there's a large fraction that is determined by non-genomic factors. And if we're going to use cohorts to explore in a holistic way the determinants of health and health outcomes, we need to take a bigger approach than just genomics and think about layering on the additional, both molecular and non-molecular layers, including upstream environmental determinants. Okay, so to address this need for holistic programs, 5-6 years ago, we set up this, what we call the SG100K cohort study here in Singapore, which is a representative population sample, 100,000 Singaporeans, men and women from across 3 Asian ethnic groups, and we are enrolling them to do deep clinical, molecular, and health outcomes exposures. And in the middle bit is a snapshot of the research phenotyping we do at baseline, which draws heavily upon UK Biobank, so that what we're doing is interoperable and we can do scaled-up analyses internationally. From the samples that we collect from participants, we've got all the sample types we think you need to do modern epidemiology, both here and thinking ahead for the future, what might be needed. And we've already started to deploy them across a number of collaborations with key partners in a really productive way that we value greatly. Okay? So that's the cohort. That's the deep phenotyping, the deep molecular phenotyping. I'm going to talk about the use case. And the use case I'm going to talk about is diabetes, which is obviously a tremendous global problem. It's increasing in all regions of the world. It affects 400 to 500 million people presently. But what's not always well appreciated is that Asian populations have an increased risk of diabetes that's not well understood. And the top left panel shows the across Asian sexes, how the risk of diabetes is about 3 times higher among South Asian people compared to European people who's living in the UK. Now, when I was a younger man, I thought, oh, it's all going to be adiposity, an increase in adiposity in Asian populations compared to Europeans, or it might have a genetic basis. And that's the middle 2 slides, showing that even when you take account of ethnicity or genetic risk, there's no take account of adiposity. Adiposity doesn't explain the increased susceptibility to diabetes. And when you look at, say, risk allele frequencies from Diagram and other GWAS-type studies, there's no systematic difference in allele frequencies between populations to account for susceptibility in Asians. So we can't find a genetic basis for it, and we're not clear what the potential behavioral and environmental determinants are. So 10 years back, And before we started SG100K, as a motivation to starting SG100K, I did an initial study with Marie Lowe and others in the audience to look at DNA methylation as a potential signature for disease pathogenesis. And the reason for focusing upon DNA methylation is it kind of integrates both genetic exposures, but also environmental and lifestyle exposures, as I'll talk to a little bit later on. And we thought that might be a good place to find the unknown. And in a small-scale study of a couple of thousand people done back in the day, we found 5 loci associated with diabetes that had risk ratios of 2, so not bad, like cigarette smoking and cardiovascular disease. But we were frustrated. We were frustrated because we didn't have the data to inform what are the upstream determinants of these methylation disturbances. We didn't have any RNA-seq data to understand the downstream consequences, and we didn't have the tools from genetics to be able to do Mendelian randomization type studies. So we called a bust. So having then created this fantastic multi-phenotype cohort, we're now able, in 2024, to do a better job and to move beyond state of the art. And I'm going to talk through the multi-omic experiment we've done to showcase how SG100K is useful as a resource for addressing complex chronic disease, and talk about our SNP methylation expression phenotype integration, data integration. And experimental design is first we start with epigenome-wide association study using a microarray to identify some CPG sites. Then from that we do a PheWAS to identify upstream determinants. We do some functional genomics to get the pathways in the middle. We do some fine mapping to say, have we got the best signals? And then we try to explore the population relevance. So 6 chunks of data analysis. Okay, epigenome-wide association, results summarized. We find 420 CPGs across 315 genomic loci associated with future diabetes, and that's a 60-fold increase on knowledge from 10 years ago. Not bad, great. Point number 1. Point number 2 then, so here's our core set of CPGs linked to diabetes. What are the upstream determinants? And there we draw down on the dense phenotypic associations we've got, including physiological and behavioral characterization. And the PheWAS results are always difficult to synthesize because you've got thousands of phenotypes potentially being measured. But what I've sucked out from is some of the key insights, and that is it's the relationship of the exposures with an unfavorable shift in DNA methylation, unfavorable marked by red, favorable by blue. And what we see is that, for example, we're identifying a lot of the behavioral and environmental exposures driving these methylation disturbances. In particular, healthy components of food, fruit intake, healthy diet, AMED, Mediterranean diet scores are protective, but unfavorable things such as intake of processed meat or visceral adiposity are strongly adverse. So we're getting into things that we're able to identify those behavioral and other characteristics that are not always measured in phenotypes or in cohorts and clinical studies that are impinging adversely upon DNA regulation. Okay. Chunk of data number 3. We've got this set of 300-odd CPGs. What do they mean in terms of disturbances of nuclear regulation? On the left-hand plot, we've got the distribution plot for methylation levels amongst our target CPGs, 300 in red, and in blue, background CPGs. And what you see, the background CPGs are typically hypo or hypermethylated, that's switched on or switched off, but the ones that are disease-associated, phenotype-associated, they're in a transition state. Some of them are on, some of them are off, and you get 50% kind of methylation on average, telling you that they're in a regulatory state. And the two right-hand panels just briefly show for the middle one, it's covariation between levels of the CPGs, the methylation marks, and on the right-hand side, covariation between the genes proximate to those CPGs, showing that there is an excess of covariation greater than expected by chance, indicating that these things are working as a set of nuclear disturbances contributing to diabetes. Okay. What other functional evaluation can we show? We can show that the methylation sites pile up in DNA, in annotated genome regulatory features, particularly enhancer regions. And as I mentioned, there's a pile-up of association with cis gene expression, and that's shown in the left-hand panel. The markers are threefold enriched for cis gene expression. And interestingly, what are the genes that are associated? It's genes that by gene set enrichment analysis are associated with insulin signaling and lipid metabolism, things that are core to the biology of diabetes. So it's compelling in terms of the biology. Okay, bucket of data number 4, 11 minutes to go. So we've got some CPGs, we've got some phenotypic disturbances, we've got a disease state. How are we going to put it into some kind of knowledge? We've got an excess of information, what's the knowledge that we're understanding? And so now we're going to try and integrate the information into a coherent set of biological pathways. And there are obviously lots of ways of doing pathway analyses, network analyses. The simple approach I'm going to show is based upon identification of using the genetic variants that influence these methylation patterns as instruments to understand the biology. So we do a GUS against the methylation marks. The GUS in cis, it shows that about 80% of the methylation marks have got some local cis-acting genetic variant that influences them. Great. And fantastically, those cis-acting genetic variants are strongly enriched for diabetes and metabolic phenotypes, further demonstrating the causal importance of these regulatory disturbances in terms of driving metabolic outcomes. But the bit that's really exciting is that these methylation marks are not just influenced by cis SNPs, they're also influenced strongly by trans SNPs, that is, SNPs on different chromosomes impacting upon methylation in trans. So we're talking about nuclear regulatory pathways influencing the methylation marks. And those trans-acting SNPs are again also strongly enriched for association with diabetes and metabolic phenotypes, part of the same causal journey. Okay. Now this is a bit of a complicated slide, but it shows why these trans SNPs are potentially interesting. It's a Manhattan plot. We've seen lots of Manhattan plots from GUS studies. But instead of being a p-value on the y-axis, what we have now is a number of phenotypes associated with the SNP. And in this case, the number of phenotypes is the number of methylation marks. And we see it's piling up. And that indicates that some SNPs are operating across multiple CPGs across the genome. They're operating as master regulatory regions. And I've just highlighted the names of 3 of them. NF-κB1, regulating 37 SNPs, its primary inhibitor, NF-κB-IE, regulating 26 CPGs, and ERG, another nuclear regulatory protein, influencing 45. And so now what I'm intimating is 2 things, that actually this CPG set is in turn regulated by a bunch of nuclear regulatory proteins, and those are predominantly coming from inflammatory pathways. And we can then put that all together as a run-through from one end to the other. We've got these trans SNPs and the eQTLs that they determine that we can show using causal inference testing are impacting upon our sentinel CPGs that we discovered as associated with diabetes. We can identify eQTMs that look like interesting gene sets, and we can use causal inference testing to show that the trans SNPs, the eQTLs, the sentinel CPGs, the cis eQTMs are linked to metabolic disease, a nice coherent end-to-end pathway. And the gene sets, I've told you about the eQTM gene sets that are linked to insulin signaling and lipid metabolism. The gene sets for the trans SNPs and the eQTLs, they're all about inflammation. So we're additionally showing how upstream inflammation is driving downstream metabolic disturbances. And the diabetes doctors in the audience, I hope, will recognize that inflammation is a core process that develops early in the pathway to diabetes. All right, now how do we then bring them into slightly more coherent biology? Well, this is a lovely bit of work done by Prof Chen, who I hope is somewhere in the audience, where he said, okay, well, some of the NF-κB influences 37, NF-κB-IE 26, and actually it turns out those are the same CPG sets. They're part of a cluster. And if we apply cluster analysis, we can pull out CPG sets that are covarying and are covarying in relation to underlying trans SNPs and the eQTLs that are driving them. And so the largest cluster on the top is full of inflammatory proteins, NF-κB, NF-κB-IE, NF-κB-IE, NF-κB-IA, and is impacting upon a bunch of important metabolic genes, ABCA, ABCG1, and CDKL1 as quick examples. There's lots of interesting unknowns in there. So, for example, SLC27A2 is a fatty acid transporter. Why that should be linked to ABCG1, ABCA1 is not known. So this is an opportunity to unravel novel biology. Okay, point number 5, 6 minutes to go, is having put together the networks, let's think about whether we've got the right markers and how we can apply this knowledge to clinical translation. The problem with the array, which was our starting point, is it only covers a fraction of the epigenome, and 3%. And taking one of our top loci, like TIC-SNP, If you look at the locus, only one of the CpGs at the locus is actually quantified by the microarray. So how can we really have a detailed understanding on risk stratification, mechanism, etc.? So we need to do better than that. And to do that, we need to do some resequencing to go from the 3% to something closer to the 100%. We did this in 2 phases. First of all, we did whole genome bisulfite sequencing. This was one of the most painful exercises I've gone through in life. Trying to do whole genome bisulfite sequencing on 500 people. I really don't recommend it. There are much better technologies now. But the outcome was, on the basis of the whole genome bisulfite sequencing, we could then delineate where the local correlation patterns are. And we say, okay, here's our top CpG from the array. What are the CpGs nearby that may be more important in driving the biology? And typically, there's about a 500 base pair interval around the lead CpG that shows some degree of correlation. So we then put that onto a Twist custom methylation panel with 114,000 markers and assayed 1,900 odd people, half who went on to develop diabetes, half who remained free from diabetes, and did high fidelity 500x sequencing. And I'm going to call out in particular Jeremy Dunsday and Justin Lee, who are heads of teams, so they're leading teams, but call out how Twist and Novogene AOT were fantastic partners in helping deliver this project at some speed. Okay, so what do we find? Well, here, for example, is resequencing of the ABCG1 locus, which was our top associated gene from the array. And what we see is, first of all, we have more markers at the ABCG1 locus than we get from the array. The array's got 1, this has got 25 to 30 associated with diabetes. We also get markers that are more strongly associated with diabetes than the index marker, which is labeled CpG 065000161. So more markers and better markers. Good for risk stratification. But also interesting in terms of mechanism, all the associated markers line up on the ABCG1 gene. And this is quite one of the useful properties of DNA methylation. Unlike SNP patterns, SNP LD is long. It's hundreds of kilobases. But DNA methylation is short and precise. And so it really does help identify, well, I propose, the causal genes involved in the biology. And beyond that, in addition, if we look within the distribution of those markers within the gene, they all pile up within enhancer regions. So the methylation resequencing is giving us more markers, better markers, pinpointing genes and identifying, giving insights into the causal mechanisms. Okay. Right. Point number 6. How we then, who cares? How does this actually help anybody? So to do that, we synthesize the data. Some of the individual CpGs, that's doing quite well. Relative risks of 2 for diabetes, that's not bad between quartiles. But if we put them all together as a methylation risk score, then potentially we might be able to get even better risk stratification. And this shows a comparison of polygenic risk scores pre the inclusion of rare genetic variants, so we can do a better polygenic risk score, it's fine, versus an array-based methylation risk score. And the array-based methylation risk score gets a relative risk of about 5 in quartile 4 compared to quartile 1. It's not bad. People on this side of the room 5-fold higher risk than the people on this side of the room. Importantly, the information between PRS and MRS is additive. They're independent data streams, which aligns to the fact that the MRS is being driven predominantly by behavioral and environmental processes and less so by underlying genetics. Interestingly, whereas polygenic risk scores don't actually differ that much between ethnicities, they do a bit, but they don't vary that much and don't account for much of the difference in disease risk between populations. Methylation risks are strongly different methylation risk scores are strongly different between populations and explain a very large fraction of the difference in diabetes risk between ethnic groups. This is an example of Indian versus Chinese. Indians in Singapore have got a 4-fold higher risk of diabetes compared to Chinese, and the methylation risk score based upon array explains 60% of that excess risk. Not bad for one panel. Okay, but I've highlighted that resequencing improves information content. And guess what? If we then apply the methylation risk score concept using a resequence locus, then our methylation risk scores become even stronger, about 2-fold stronger, even with the relatively limited experimentation we've done. So that the Q4 to Q1 risk now becomes 9-fold higher risk compared to the group over there. That's quite important in terms of stratified medicine, potentially. And It's independent of genetics, and it's additive on top of things like classical risk factors such as adiposity or prediabetes. So in a subgroup analysis, we can take the obese without prediabetes, and even in that group, we're getting obese but not pre-diabetic, 5-fold, 1-fold difference in risk. It's fantastic, we think. Okay, so with 54 seconds to go, Jeff, a conclusion slide. I have tried to demonstrate the power of multi-omic investigations for unraveling complex multifactorial disease, using, in this case, the examples of SNP methylation expression phenotypes. But the molecular layers are much richer, and there's fantastic stuff coming out of the proteomic profiling, metabolomic profiling, etc. The conclusion I've got is we find 300-odd markers predicting diabetes. We're able to use that knowledge to understand the behavioral markers driving disease risk in addition to the genetic risk factors. We're able to generate meaningful risk prediction that is additive to genetic risk and helps explain risk between populations. We're able to generate coherent biological pathways with novel biological insights about nuclear regulation underpinning future diabetes. And I've demonstrated the tremendous potential for resequencing-based exploration of the epigenome, whether it's using the Twist methyl-epic type platform, methyl-array type platform, or whether it's using long-read type platforms such as ONT or PacBio to get whole genome methylation data. And I think it's going to be really exciting as we move forward with this cohort work, but also look forward to seeing how other people build upon our knowledge. Thank you for your attention.
Well, thank you, John. Thank you for talking very fast and for staying on time, and also for highlighting the ability for human cohort data to enable us to derive multimodality risk scores and also to dive in deeper into the biology of type 2 diabetes. So for our next speaker, we have Professor Zhengming Chen, who was an elected member of the Academia Europaea and an inaugural Richard Pito Chair of Professor of Epidemiology at Nuffield Department of Public Health at the University of Oxford. In 2003, he established the China Kadoorie Biobank, and I believe he's going to use the China Kadoorie Biobank to tell us about harnessing the power of proteomics and biobanks to advance precision health. Thank you, Zhengming.
Good afternoon. Thanks very much for the introduction. I'm delighted to be here to share with you our recent work on proteomics. As you all know, proteins are the building block of our life, and most drugs target protein. So with the recent development of technology, so we're now entering post-genomic era. So it is a game change, will be transformative, and so somehow the epidemiologist will be at the forefront of biomedical research. So this will greatly advance the development of precision medicine. So for most common complex disease, despite recent development, our ability to prevent, to treat, are still somehow limited. So as you can see here, there are still very large, well-documented, unexplained variations in disease mortality between different populations, as well as between different regions within the same population. So this shows the two very common cancer in China. You have red hotspots where the mortality rates are 5 to 10-fold higher than the neighboring regions for reasons we still don't understand after 30, 40 years. So in a way, this reflects sort of an incomplete understanding of human biology, disease etiology, as well as a very complex interplays between genes, environment, nature, and nurture. And so this is important, and because between different populations, the interaction may operate very differently because of the different disease rate, different lifestyle factors, and also different genetic architectures. So what we need is really to study diverse populations. So for the development of precision medicine, we need much better evidence base for better understanding of human biology, disease etiology, much better tools to predict disease, to diagnose disease, and to detect disease. And also we need more medicines, new medicines, to treat perhaps sort of a more targeted treatment. And also, to categorize patients for more targeted treatment with a much better efficacy and safety profiles. So I think to develop this, cohort study is essential, especially if you want to study non-genetic factors. This is really important, proteomics, metabolomics, especially if you want to study the interactions, gene-environment interactions, especially if you want to study multiple disease simultaneously. The only thing can do is big biobank studies. So 20 years ago, we started this huge endeavor, China Taiwan Biobank, which is long-term collaborative studies between Oxford, so I'm the UK PR, and with Professor Lin Yen-Feng in China. So this is the largest kind of established biobank ever established in non-European populations. It is very large, with over 500,000 people recruited from across 10 different carefully selected areas, from freezing north in Harbin to tropical south, from coastal city to inland and rural areas, with very different disease patterns, different stages of economic development, but otherwise everything standardized. And the study is very deep, with extensive information collected by questionnaire, by physical measurements, and increasing with assays of biological samples, whole genome sequence, proteomics, metabolomics. This is still sort of increasing. And with a very detailed collection of a wide range of disease outcomes as well. And also it's very long term. Now we are celebrating 20 years of Taiwan Biobank, but still at least have another 20 years to go. And also this data is opened up several years ago, and this will be further enhanced by development of a cloud-based system to facilitate direct access like UK Biobank as a genetic information. So this is a huge undertaking. A big study is not a small study made big. In order for the study to be successful, you have to have a very good study design, quality assurance, infrastructures, training, monitoring as well. So this is a book I edited, which is available on Amazon as a self-promotion. If you're interested, hopefully that will be useful for countries that try to set up different studies, new studies. One of the most important aspects is actually the whole study, no paper. It was managed, supported by over 100 different bespoke IT systems as well. So this just shows kind of framework of all the ICT system involved to support all aspects, that not only data collection, but also sample management, sample movement, storage, and as well. So this is very complex, but this made study very, very smooth, extremely high quality, and very efficient. So when I say high quality, what's evidence? First thing, there's no missing values, no log errors, and a very complete sample collection. Only 96 samples not collected, with no sample loss, damage, or unintended source. Very complete follow-up. After almost 20 years, less than 1% lost to follow-up. We're not only able to collect mortality, but also importantly, any episodes of hospitalization. Within about 18 years, almost 100,000 deaths, more than 2.5 million episodes of hospitalization for all human conditions. Not only cancer, diabetes, stroke, but also dementia, Alzheimer's disease, schizophrenia, depression, even snake bite in some part of area, earthquake deaths in one regions as well. But also the disease outcome is very well categorized. Give you one example, 95% of stroke diagnosis based on imaging, reflect the quality of the hospital care. But we also spend a lot of time trying to further enhance disease phenotyping by retrieve original medical record for about 10 major diseases. This is quite important to subphenotype to enhance the future research. We also done 3 repeat assessment with a very high response rate, but also with a lot of enhancements, including new samples like fecal samples, saliva samples as well, to prepare for the future research. So over the last 10, 15 years, we have undertaken a lot of research. We also have a lot of collaboration, open access. I mean, yes, compared with the UK Biobank, we're far behind, but we managed to publish over 500 publications. With some ended up in the best medical journals. Some actually just confirm what we know, but with a much better precision, some old questions. Some actually challenges a common wisdom, for example, whether moderate drinking is beneficial. So we got the first best evidence. I'm not going to say what's the results, because I don't want to spoil the dinner this evening as well. And also a lot of research are hypothesis-free, so discovery research. So this is enhanced, improved by arrival of the genetic proteomic, which is I'm going to mention. So what we can discuss is a small pilot study with 4,000 participants. So we measured 2 platforms, Olink, SomaScan. So now we have over 30 active projects, but this could be easily increased by 10-fold, because basically each of the 10 proteins, that could be a PhD project. So that's actually the power of this kind of biomarks, which cover a wide range of topics, some sort of GWAS, risk prediction, drug target, biological aging as well. And also, this is the largest direct comparison of 2 platforms as well. So we have published, submitted over 15 papers. This is just a selection of some of the papers published and accepted as well. So in the interest of time, I'm not going to go through all of them, just mention some of the highlights. So this is almost like takeaway message. So first thing, the levels of plus proteins are associated with a wide range of genetic, non-genetic, and environmental factors. This is quite important, like ambient temperature, fasting time. And also, proteins greatly outperform genetic risk scores for many diseases. I will show some of the results. In GWAS, in Chinese, 40% of PKTLs did not seem to overlap with those Europeans. This emphasizes the need to study diverse population for major discovery. The genetic analysis, Mendelianization, colocalization help to clarify the causality, and also to identify many drug targets for a wide range of disease. And also use the proteins, you can develop a really powerful biologic aging to predict not only the age-related traits, but also many disease mortality. I'll show some results. And also, with Olink SomaScan, if you got money, the best thing to do both. So that's my view. So I think the analysis has become very sophisticated. So not only Cox model logistic, but involve probably 7-8 layers analysis in any investigation. So increasingly, we also start to use machine learning, artificial intelligence, downstream analysis, knockout models, try to nail down not just whether association, is this causal, is this real, is this potentially drug targets as well, what's the mechanism? So that's the key results, take-home message, and then I'm going to run through very quickly. I'm not going to go through specific detail, just give you a flavor of the key study findings. So the first thing you want to see actually how proteins are affected by different things. So this is called, we call atlas, or exposomes. So this is what we did with Olink SomaScan. And at the bottom, this was selected. We've got many hundred different variables, so we've done that systematically. Age, sex, and the lifestyle factors, environment factors, I mentioned the temperature, air pollution, etc., and also physical measurements, reproductive patterns. As you can see, a lot of hits, some positives, inverse, but overall, between 2 platforms, very similar. So there's loads of them. But interestingly, for diet, hardly any, to our surprise. And then we can zoom in to look at the specific. This is, again, selected exposures, regular alcohol drinking, current smoke, random blood glucose, fasting time, outdoor temperature, blood pressure, prevalent diabetes, HBV, antigenemic. So there are a lot of hits as well. But each hits you need to zoom in, actually, try to understand. So I'll just show the patterns, the number of hits. This is a very typical analysis volcano plot. So each one is like one biomarker, protein biomarker. And then for adiposity, so we use the 4 different traits, BMI, percentage body fat, waist circumference, waist-hip ratio, massive numbers. So this is a SomaScan, Olink, very similar. Obvious questions, is this causal? So we use the Mendelian anomaly to confirm many hits are causal. So the adiposity causally affects the level of protein. But using the pQTL, we can also assess whether some proteins affect levels of adiposity like GLP-1, which is a wonder drug to treat obesity. So we identify, I don't have time to show, we identify probably 30 such proteins. This is quite potentially druggable as well. And then for all-cause mortality, again, 2 platforms, very similar patterns. Putting together, we have almost 1,000 protein hits. They are strongly associated with all-cause mortality based on relatively small numbers. But this is after adjustment for all the confoundings, for multiple testing as well. And then you can use these proteins to improve the prediction of the all-cause mortality very substantially. And also they are better than conventional risk factors as well. And then for other disease, chronic renal disease, chronic liver disease, we have 1,200 hits, chronic liver disease, 500. Of course, does not necessarily mean they are causal. We need genetic. So this is, again, ischemic heart disease. Luckily, this is a nested case cohort study. So we have 2,000 IGD cases, 2,000 controls. We're able to do very well-powered analysis. SomaScan, Olink, a lot of hits, and several hundred. And then they improve the risk prediction. As you can see on the top, conventional risk factors, C-status 0.845, PGS 0.5, very weak. Proteins, much better. But important things, if you add them together, especially add protein to conventional risk factors, substantially improves the risk prediction as well. So this is a ROC curve, as you can see. And GWAS analysis, the paper, revised paper will be submitted. This is again based on Olink, because another SomaScan paper is coming. So even with the 4,000, we identify a lot of cis-pQTLs and the trans-pQTLs. So overall, Chinese, Europeans, very similar genetic architecture. But if you look at the detail, there's some difference. Some actually, we got cis-pQTL, but not in Europeans. Obvious one with alcohol, there's a fresher genes, ALDH2. And also some lead variant within struct genes in Kaduri, but not in UKB4, 155 proteins. And also even for a lot of the proteins, over 40% pQTL, they appear to be distinct from those identified in Europeans. So this is, if you study different population, you will greatly improve the sort of novel discoveries as well. And then use the pQTL, especially cis-pQTL, you can do a lot of kind of genetic analysis. So this is one paper will be submitted very soon. So this is actually using cis-pQTL for drug target discovery. So again, as you can see, with the selection, instrument variables and conduct 2-sample MI, and also co-localization, and replicate between different platforms, and also replicate with the UK Biobank. And then we identify 32 potential drug targets. And then further downstream analysis of PWAS, knockout models, look at all the libraries. So eventually we found this, many of them actually complete novel, potentially drug targets. Some actually, there's already some drug development, but for complete different release, so there's a potential for repurposing. I mean, here just show the Olink SomaScan, the 32 proteins. Some are overlapped between 2 platforms. I just want to highlight this very well-known drug target, PCSK9, which is replicated as a positive control, demonstrates the power of this kind of approach for drug discovery. My last part is biological aging. We have some talks today. This is a very hot topic. I think until now, there's a lot of aging clock available, but mainly based on the methylation. But our question is whether use a protein can improve the kind of biological aging prediction. The answer is definitely yes. They perform far better than other available aging clock. So this is a paper published just 2 weeks ago in Nature Medicine. So in terms of analysis, very standard, using the UK Biobank to develop the model, 70% sort of test case, and then 30% training case data, another 30% test, and then replicate the model in 2 cohort study, China Kadura Finjin as well. And then, as you can see, the protein age predict really remarkably across 3 populations. Your chronological age very nicely. And also, you can calculate the difference between biological age and also your calendar age to derive what we call the protein age gap. If someone like 50, your real biological age could be 45 or 65. So this is what we show, the age distribution, very well validated, male, female, UKB, CKB, FINGene, and also different ethnicity, white, Asian, black, mixed, other, very nice performance. And also even across 10 different regions in Taiwan Biobank, wonderful sort of consistency as well. But the important things is not just predict your age, but this age clock also predict reliably 20 age-related traits like muscle strength, bone density, your facial appearance, and also telomere length as well. But most importantly, they also predict multiple disease, like over 20 different conditions as well, all-cause mortality, morbidity, as you can see. When we saw the result, we were surprised, too good to be true. You know, why it's just simple test, 200 proteins or even 20 proteins, you can see. Heart disease, stroke, ischemic stroke, COPD, liver disease, kidney disease, all-cause dementia, Alzheimer's disease as well. I don't show everything. But also very consistently in Taiwan Biobank, the number a bit small, but the patterns are very clear. All-cause mortality, stroke, ischemic stroke, heart disease, type 2 diabetes as well. But this is the really beginning. There still is a lot of opportunity for further research. For example, we want to develop sex-specific aging clock, organ-specific clock. Because at the end of the day, many people die not from all organ failure, maybe one sort of heart disease or some cancer. So this is important. And also want to incorporate other like methylation, metabolomics data, try to further enhance. And also there's a potential for interventional study use aging clock as a very nice kind of instrument or intermediate biomarker as well. And also there's a depth scope for clinical translation as well. So I hope give you some sort of a high-level description of the potential of the proteomic profile in biobank. They will greatly improve the understanding of human biology, disease etiology, identify novel kind of drug targets, improve the risk prediction, early diagnosis, mechanism linking lifestyle with disease, molecular basis of genetic variants associated with disease, patient stratification, drug response as well. So this is a game change, especially if you're able to incorporate this together with genetic. This will be totally transformative. Before the biobank, the key challenge opportunity is to turn sample into data. For us, we want to scale up to 50,000, ultimately, or 500,000. And so this will be very, very exciting development. Finally, I'd like to thank all the collaborator founders. This is a huge collective effort. So I'm going to stop here. Thank you very much for your attention.
Zhengming, that was terrific, and it's maybe it's obvious, but one of the purposes of biobanks is to store samples, and if you don't convert samples to data, then you're not fulfilling your mission. So next up, our last speaker in today's session is Dr. Yen-Feng Lin, who is a clinician scientist, psychiatrist, and epidemiologist. He's at the Taiwan National Health Research Institute, where he runs the Taiwan Biobank, which was initiated in 2012. And today he's going to talk to us about genetic analysis of quantitative traits in the Taiwan Biobank.
Many thanks to Dr. Ginsburg for the introduction. It's really my honor to be here as a junior investigator. I'm very grateful for this opportunity to present here today. And I'm going to talk about our work on genetic analysis of quantitative traits in the Taiwan Biobank and the comparisons among Taiwan Biobank and Biobank Japan and the UK Biobank, and also demonstrating the necessity and the importance of using data from diverse populations. First of all, I'd like to give a quick overview and introduction of the Taiwan Biobank. And the TWB is a community-based prospective cohort study of the Taiwanese population with multi-omics genomic data and longitudinal phenotypic and environmental measures. And since 2012, The cohort has been enrolling men and women aged from 30 to 70 with no prior diagnosis of cancer from more than 30 recruitment sites across Taiwan, distributed based on population density of different counties and cities with an expected target sample size of 200,000 by the end of 2025. And at baseline, each participant undergoes physical exams and an interview with a well-trained interviewer to report a wide range of phenotypes and health-related information in a structured questionnaire. Here I listed the phenotypes by category. There are demographics, behavioral, environmental, diet, anthropometric, and the medical imaging phenotypes. And health condition-related phenotypes are self-reported diseases, family history, and results of physical examinations. Blood and urine samples are also collected for biomarker testing and the generation of genomic, methylation, and metabolomic data. At each follow-up visit, a repeated assessment was carried out for phenotypes measured at baseline. A total of more than 1,000 phenotypes have been measured. And in addition to self-reported medical conditions, disease and health information can be obtained through linkage to Taiwan's National Health Insurance Research Database. And also, there are many other additional databases that cover specific health-related outcomes in the Taiwanese population, such as the Taiwan Cancer Registry and Cause of Death Registry. And the Taiwan's National Health Insurance is a single-payer compulsory insurance program instituted in 1995 for all citizens in Taiwan, with a coverage rate of over 99%. So there are comprehensive medical claim and health insurance data available upon application. But cross-linkage between TWB and these healthcare databases currently only permits on-site analysis with a separate study-specific IRB approval. So efforts are underway to integrate health-related records from across these databases with the TWB community-based phenotype collection. And here's the distribution of age, sex, and education level of study participants at baseline. And more females than males were enrolled in TWB. Age of the participants was evenly distributed across every 10-year bracket, and 75% of the participants had a high school degree or higher. And the most common self-reported disease is peptic ulcer, with a prevalence of around 15%. The followings are GERD, hypertension, hyperlipidemia, and kidney stone. But one noticeable issue of TWB is several self-reported common diseases showed an in-sample prevalence lower than that in the general adult population. For example, the in-sample prevalence of hypertension is 12.5%, while the prevalence is 26% in the general Taiwanese population. So this may reflect biases from self-report or similar to what was observed in the UK Biobank of a healthy volunteer bias. And multi-omics data were generated for all or subsets of the participants, including genome-wide SNP genotyping, whole genome sequencing, HLA typing, DNA methylation, and blood metabolome. And the population structure of TWB is highly homogeneous, comprising Taiwanese individuals of predominantly Han Chinese descent. In the left PCA plot, we projected the TW samples onto 1,000 genome data showed tight clustering with the East Asian superpopulation. And in the right plot, among the 1,000 genome East Asian samples, TWB subjects are clustered with the two Han Chinese populations, which are CHB and CHS. Okay. The main results I'm going to report are findings from around 100,000 individuals across 36 human quantitative traits in the Taiwan Biobank, which has been published in Cell Genomics last year. And genotyping for Taiwan Biobank was performed using two different customized genome-wide arrays. In the sample of our analysis, around 27,000 participants genotyped on the TWB1 array and 75,000 genotyped on the TWB2 array. And the 75,000 individuals using TWB2 were divided into two subsamples. 65,000 were used as the discovery batch 2, and the other 10,000 were used for polygenic risk prediction, validation, and testing. We performed GWAS for 36 quantitative traits, which are listed in the right panel. We also compared our results with those from Biobank Japan and the UK Biobank. And the quantitative phenotypes in our analysis include anthropometric traits, bone density, cardiovascular traits, blood cell measures, serum biochemical markers, and the lung function measurements. And after stringent QC, we performed the GWAS on 36 quantitative traits with imputed genotype data. We used Regeny, a two-step whole genome regression method for genetic association tests that account for sample relatedness and the population structure to perform association analysis on the two discovery batches separately. And we used linear regression in Regeny for association testing, controlling for age, sex, and the top 20 principal components. And LD score regression intercept and lambda GC showed that there was negligible inflation due to population stratification. All traits had high between batch genetic correlation estimates. So then we meta-analyzed the GWAS results across the two batches. And this plot summarizes results of the meta-analysis. Each layer of the plot represents a single trait with traits within the same category grouped by the same color. And each dot represents a genome-wide significant hit. The most pleiotropic genes identified in the study are annotated at the bottom. So we meta-analyzed the discovery batch 1 and 2 GWAS using an IVW fixed effect approach. We identified 1,986 independent genome-wide significant loci across the 36 traits, among which 1,300 loci survived Bonferroni correction for the number of traits tested. The number of genome-wide significant loci per trait ranged from 2 for FEV1 to FVC ratio to 229 for height. And all summary statistics are publicly available on GWAS catalog. We also estimated the SNP-based heritability for each trait. And we estimated pairwise genetic correlation between traits and identified clusters of highly genetically correlated traits. For example, body fat rate, BMI, body weight, waist circumference, hip circumference, and waist-hip ratio appear to be highly genetically correlated. And we implemented a summary states-based version of SUSY for the fine mapping analysis. And of the 1,900 loci, around 300 failed to identify a reliable, credible sets that passed QC threshold. And the remaining 1,600 were fine mapped to a total of 1,900 credible sets, each representing an independent association. And out of the 1,900 credible sets, 232 were mapped to a single variant with posterior inclusion probability greater than 95%, among which 24 were missense variants. So here we list the 24 fine-mapped missense variants with PIP greater than 95%. And all fine-mapped missense variants with the corresponding GWAS available in Biobank Japan or UK Biobank were replicated with the same direction of effect and the study-wide significance with a p-value smaller than 0.05/15, which is 0.003. And these putative causal missense variants not only replicated previous findings, but also represented novel causal variants. For example, the RREB1 gene for bone marrow density. And some of these high-PIP missense variants were also highly pleiotropic. For example, the SLC10A1 gene is associated with gamma GT, LDL cholesterol, and total cholesterol. This suggests its pleiotropic role in multiple complex traits. And prior studies have shown that complex traits are genetically correlated at different levels between East Asian and European populations. But the genetic overlap within East Asian populations has not been characterized. So leveraging existing GWAS summary statistics from Biobank Japan and the UK Biobank, we investigated the comparative genetic architecture of quantitative traits within East Asians and comparing TWB and BBJ and the cross-population between East Asian and the European population, which are TWB versus UKBB and BBJ versus UKBB. Among the 21 traits for which GWAS were available across the 3 biobanks, the SNP-based heritability estimates in TWB were comparable with those in UKBB, but consistently higher than the heritability estimates in BBJ. So a possible explanation might be that both TWB and UKBB are community-based, while BBJ is hospital-based, in which biomarkers measurements may be affected by health conditions and the medications used for the patient sample in hospitals. We also compare within East Asian and cross-population genetic correlations. Within East Asia, TWB-BBJ genetic correlation estimates were in general higher than cross-population genetic correlations. But despite these differences, we know that all within and cross-population genetic correlation estimates were high with a median of 0.9 something. And taken together, these results suggested that the genetic architecture for the quantitative traits examined here was largely consistent across East Asian and European populations. To maximize the power for genetic discovery in East Asian populations, we also meta-analyzed the GWAS from TWB and BBJ for 23 traits with data available in both biobanks. We report a signal in the meta-analysis as novel if none of the variants within the locus reached genome-wide significance in previously reported BBJ or UKBB GWAS. We identified a total of 3,382 loci associated with the 23 traits, among which 665 were novel. And for the other 13 traits for which BBJ GWAS were not available, we identified an additional 400 genome-wide significant loci using TWB samples only, among which 300 were novel. And we found many of the associated loci were highly pleiotropic. For example, the TRPS1 gene was associated with 14 traits spanning the anthropometric, bone density, hematological, and metabolic categories. And the mechanism underlying this pleiotropic association will need to be further studied. And with biomarker GWAS results available, we assessed the clinical utility of biomarker GWAS for disease risk prediction. We examined whether point risk scores of biomarkers can be used to predict the risk of common complex disease. We applied PRCSX, a method for multi-ancestry polygenic prediction, to integrate the GWAS summary states of East Asian and European ancestry and calculate both an EAS-specific and an EUR-specific PRS for each biomarker, and then combine the 2 population-specific PRS to be one final cross-population PRS. We then predicted 5 complex phenotypes, including obesity, overweight, hypertension, hyperlipidemia, and type 2 diabetes, in a held-out TWB sample of around 10,000 individuals using a linear combination of PRS from 1 or more biomarkers, controlling for age, sex, and top 20 principal components. And we found biomarker PRS were significantly associated with disease status, it can explain around 10% of the variation for obesity, overweight, and hypertension, and 6% of the variation for type 2 diabetes, and 4% of the variation for hyperlipidemia on the liability scale. And as a comparison, we also predicted type 2 diabetes using the largest to-date disease GWAS in East Asia with 280,000 individuals, and in European with a sample size of 900,000. And while the disease GWAS represented much larger sample sizes, a combination of only 2 biomarker PRS, which are HbA1c and BMI, was able to explain approximately half of the liability variation compared with the PRS derived from large-scale GWAS of type 2 diabetes diagnosis. So in summary, we performed genome-wide analysis on 100,000 community-based TWB participants across 36 human complex traits. We found that the genetic architecture for these quantitative traits was largely consistent within EAS and between EAS and EUR populations. And integrating TWB and BBJ GWAS identified a total of 3,800 genetic loci, among which around 1,000 had not been reported in previous biobank GWAS. We also fine-mapped over 200 association signals to a single variant with PIP greater than 95% and identified 24 putative causal missense variants. We also demonstrated the potential utility of biomarker GWAS in predicting disease risk and the promise of multi-trait cross-population polygenic prediction. And next, I'd like to present an example of how I have applied the results from this biobank GWAS to my pharmacogenetic studies. One of my research interests is to explore the possibility to predict treatment response of antidepressant for major depressive disorder. So antidepressant response is a complex polygenic phenotype. The largest GWAS to date with a very limited sample size of 4,000 identify no genome-wide significant association, but polygenic analysis found its genetic correlation with other phenotypes. So people started to consider the potential of polygenic scores to be predictors for antidepressant response. But one single polygenic score is not enough. We don't have large-scale GWAS for treatment response to be the discovery data for calculating polygenic scores. And we have already known that antidepressant treatment response is very complex. It's influenced by many different traits. So a possible solution is to use multiple polygenic scores for outcome prediction. So we can try to build a prediction model by combining many polygenic scores for phenotypes that may be related to antidepressant response to see if this can increase the predictive power. In this pilot study, we tried to apply a multi-polygenic score approach to predicting treatment outcome of SSRI antidepressants for patients with major depression. We used data from 2 independent Taiwanese cohorts. All participants were diagnosed as major depression and received SSRI treatment. The Hamilton Rating Scale for depression was used to assess the severity of depressive symptoms at baseline and during follow-ups. And we gathered summary statistics from about 200 published GWAS, primarily from East Asian and European ancestries, including our Taiwan Biobank GWAS results, to calculate polygenic scores for 108 traits potentially related to depression and antidepressant treatment outcomes. And these traits include psychiatric disorders, substance use, psychological and behavioral traits, brain imaging and neurological disorders, sleep traits, inflammatory biomarkers, medical diseases, and medical measurements. We then developed prediction models for percentage improvement of the depression rating scale at week 4 using different model selection methods compared to the basic model that includes only demographic and clinical predictors. The model incorporating multiple polygenic scores can increase the R-square from 0.097 to 0.172. And we developed prediction models for antidepressant response at week 4, defined as more than 50% reduction in the rating score for depression, and remission at week 8, defined as more than 75% reduction in the rating score. And all machine learning algorithms performed moderately to predict antidepressant response at week 4, with an AUC of around 0.6, which is not very good, but it's kind of acceptable in a small pilot study. And for predicting remission at week 8, we included improvement percentage at week 4 as an additional predictor. And we have AUC greater than 0.9. And we use the same method to predict treatment response of low-dose ketamine infusion in treatment-resistant depression. And so ketamine is a new option for those who don't respond well to conventional antidepressant, and it can rapidly alleviate depressive symptoms, often achieving optimal results by the second day after administration. So we use the percentage of improvement on the second day after treatment as an indicator to evaluate the therapeutic effects. And polygenic scores of inflammatory biomarkers, Alzheimer's disease, obesity-related traits, and childhood trauma were selected by more than 3 of the 4 different predictor selection methods. It is noteworthy that in this case, the correlation between the polygenic scores of inflammatory biomarkers like CRP or interleukin and improvement in depression, the correlation is positive, which means that individuals more prone to inflammation tend to respond better to ketamine treatment for depression. And this direction of polygenic score association is opposite to the treatment effects observed with conventional SSRI antidepressant. And we use different dimension reduction and the machine learning methods to build prediction model for ketamine treatment outcomes. The R-square increased from 0.019 for the basic model to 0.149 for the multi-polygenic score model, and the AUC is around 0.7 for predicting responder versus non-responders. Okay, so the above is my report on conducting GWAS using Taiwan Biobank data and how I apply the GWAS results to secondary analysis. And we hope to share our results and also collaborate with other biobanks and get access to more data to apply these GWAS results to further secondary analysis. For those who are interested in using TW data, our GWAS summary states are public available. And if you are interested in individual phenotypic and genotypic raw data, they are available upon application for research purposes. So researchers will need to submit an application, including a research proposal and an IRB approval. And for researchers from other countries outside of Taiwan, an additional international data transfer agreement will be needed. Okay, so lastly, I would like to thank all the participants, the staff, and the management team and leadership of Taiwan Biobank. And I'd also like to acknowledge the contribution and efforts of my lab members and our collaborators. And thank you all for listening, and any comments or feedback would be appreciated. Thank you.
Not 100% sure how much time we have, but I think we have close to 15 or potentially even 20 minutes. 20 minutes, it looks like. So first, I just want to remind everybody that if you are interested in asking questions, use the Pigeonhole app, pigeonhole.at, password PIC24. You can ask original questions or vote on questions that somebody else has asked. And I see some questions are coming in. But I thought I would, since I'm the moderator, I have power to ask my own questions first. And we've had 2 really great sessions on biobanks across the world today, and particularly in this session, seeing information coming in on proteomics and methylation and GWAS data from very homogeneous populations. I can imagine there's a need for bringing biobanks together for validation, for increasing sample size and other facets. So with my IACC hat on, I wanted to ask this group what you see as the key steps and strategies in developing a network of international biobanks that could effectively carry out research to improve both discovery, validation, and clinical translation. So I'll ask everyone to respond to that, please.
I guess I'm closest. Well, I mean, the first part of the question is somebody's got to collect the data in the samples. And unless somebody does that, like Zhengming 20 years ago, or Taiwan Biobank, or we do it, you're never going to get anywhere. So the fundamental step is to have the courage and enthusiasm to say, this is worth doing. And I hope some of the presentations that you've seen today persuade you that there are fantastic things to reveal and that will help the human condition. And putting together a cohort study, it's tough. It takes 5 years, 10 years. But it's not impossible. All the tools you need there's lots of advice. We benefit from a lot of advice from others. We're happy to give advice to colleagues who want to start up. So that's one foundational thing. Then how to build a network. I think in general as scientists, we've all been really pleased with the sort of the 20 years of GWAS experience of actually how productive and informative it can be to collaborate. Taiwan Biobank, nice talk on how to bring together Asian biobanks for that purpose. I rarely meet anybody who's not fundamentally really enthusiastic to collaborate now. So I think the ground is all there. What can leadership team do to further facilitate that? I think the key things are putting in place good infrastructure, good governance frameworks to say, these are platforms that are robust and work. These are data tools that we can share across environments. These are the governance frameworks that allow us to work across countries. And then the oil, the funding to say, okay, let's pump prime here and there to get a use case progressed, because one step leads to another step leads to a long journey. 3 thoughts.
Great, thank you, Zhengming.
Yeah, I want to just echo what just described. I think my observation is that there's too much data, too much high-quality data. So the question is whether they need new biobank. It's a very long-term, substantial investment. So that's the first thing people need to consider. I mean, another thing is for the existing biobank, there's so much you can do discovery research. I just mentioned protein. One protein, basically one PhD, you have 10,000 proteins. That's a scale of the potential. So I think it's really important for existing biobank to open. You have to open in order to have a position in the worldwide kind of a community. And you have to share. So this is really important. So this is, we try to embrace all the available data, because for publication, the standard getting high. You have to do, validation, combined analysis. So I think with IGC, it's a wonderful platform. So it has a great bring together. But I think in terms of future research questions you highlighted, I think I'm not quite sure how to do that, because outside IGCC, there's a lot of collaboration. We contribute data to 60, 70 consortiums. So one thing is actually in future try to bring them over here for the conference, identify a few areas so we can work close together. I mean, another thing is to identify a set of questions this community can assess while other cannot. I don't know what they are, but definitely this is quite important. So we'll not just share the methodologies, findings, actually how best we can work together to answer fundamental scientific questions, but bring together different cohorts. Not everything, but something definitely can be achieved. So that's my view.
So question-specific cohort assembly.
Okay, so in my view, I think the most important step to do is to expand the current and existing collaboration to more regions, especially for those underrepresented populations. Yeah, because I think the biggest challenge might be from the different data sharing policies across different countries. I think this issue might be minor for countries with mature biobank systems, but for those just starting, data sharing and data use may be a critical barriers to international collaboration. From our scientific perspective, we aim to collect more data to foster more collaborations. But for experts in ethics or laws, or for the general public, they don't fully understand and appreciate the potential benefits that the scientific advancement can bring. And so, for example, they have many concerns, and these concerns may lead to numerous restrictions on data use and data sharing. So, for example, some people are very sensitive about genetic data. They fear it could lead to discrimination or like sorting humans into categories of better or worse. And many biobanks are funded by the government, and some may question why the government has access to so much of my data and why it's allowed to share with others. And when it comes to international data transfer, some people may worried about it's like handing over some national important data to other countries and posing national security risk. And in many cases, collaboration between academia and industry is necessary to produce more valuable or expensive data, or to apply the research outcomes to medical practice. Yeah, but some may question whether the government is using national resources to benefit specific companies. Yeah, so all of this seem unreasonable to us, but yeah, it's like, because we know that any progress may also bring some relative risks, but it's like we cannot stop people driving just because some may violate traffic laws, right? So I think the most important thing is that we have to explain and communicate with the public to let people know the potential benefits of doing this, and explain what we are doing, and let people know how we strictly monitor data security, to explain what the identification is and how we follow research ethics. Yeah, so because we cannot force all countries follow the same standards. Yeah, but if more people understand what we are doing, then if there is a universal demand, then it will naturally lead changes in policy. Yeah, that's my opinion.
Well, thank you all for those insights. Actually, a number of questions that came up in this morning's session you addressed that really help biobanks from low resource settings and others that want to engage with larger communities of biobanking leadership. So, and you also touched on a number of issues that are coming in from the audience questions. So I feel like we should dive into some of those questions. And let's see, one of them is related to sample size. Oops, let me try that again. There we go. And it really has to do with the questions around hypothesis, non-hypothesis driven research, but more discovery research, hypothesis generating research. How do you know how many samples you need for proteomics, for methylation, even for GWAS studies? Can you elaborate on that?
So maybe I start very briefly. I think we don't really calculate sample size. The bigger, the better, but with the constraints of kind of a funding level available. But even with the proteomics, there's a different type of research. One is a purely hypothesis-free discovery. So what we have done is adjust for multiple testing, whether it's FDR or whether Bonferroni. So sometimes it could be quite conservative. But you probably still end up with many, many hits. They'll keep you busy for many, many years. So it doesn't really matter if you've got reasonable good size. For the genetic, there's a good sort of understanding. If you can do 50,000 with a good genetic data, that will probably help you to discover most, not all, the PQTRs. So that's actually quite important to know for the genetic discovery, PQTR, transcysts as well. But for the hypothesis testing, you can work on single proteins, or 20 proteins they are involved in inflammation, lipid metabolism. So that will actually, you don't need to worry other things that improve the power. So there's different ways of doing things. One is increase size, but also depends on different questions, what sort of number you can analyze.
John?
Okay, I mean, it's a recurring question in all epidemiology and cohort studies, isn't it? How big is enough? As Zhengming says, the more sample size you've got, the more power you've got. But let's reframe it. If you're going to do a study, the key thing about doing a study is making sure you've got a unique selling point. And the unique selling point may be larger sample size than UK Biobank, that's great. Or it may be a small but really an interesting case cohort that's got an interesting disease progression or treatment response. So rather than phrasing things along the sort of simple lines of how big is enough, I'd phrase it along the lines of, is your study going to make a difference to global human knowledge, which might mean bigger along a similar design, or might mean smaller but a different design.
Thank you. Do you want to answer that one too?
So I would like to echo the two other speakers. So in general, the bigger would be better. Yeah, because the concept is a little bit different from traditional study design. Because what we have been doing is to find strategies to make best use of what we have. Yeah, it depends what we have, and depends on what the research question is. Yeah.
Thank you. Let's move on also to another general question about biobanking strategy. And this one is asking about whether we really need to continue to collect data on adults, or should we focus any future biobanking efforts on underrepresented populations and also pediatric populations who are largely underrepresented or unrepresented in most cohort studies? Anyone want to take that on? Anyway. There we go.
I think there's no conflict between these two. Yeah, we can do both.
Okay, that's a good question.
Okay. I think really important to study underrepresentative samples, but also for the biobank, you need to be careful whether the study design, whether it's a prospective or whether it's a disease-specific sort of case control. Because for the prospect study, if you cannot follow them up for 10, 15, 20 years, be careful whether you do it. Otherwise, after all the effort, a lot of participants get lost. So this is quite important. Another key thing is, instead of building new cohort study, you try to take advantage of the data available, try to translate study findings into different populations, try to follow with a more detailed analysis, disease-specific things. So this is quite important, rather than start from scratch, because we have done 20 years. Until now, we just start to see some real nice stuff emerging. So it's a very long-term game.
No, I think it's a great point that, particularly with pediatric populations, you need to finish what you start. And if you don't have the funding, you don't want to have a failed effort. That's going to disappoint everybody. Let's also move on to another general question, if I can find it here. There's one, you're seeing questions that I'm not seeing, but I guess the question about the utility of the electronic health record. Is that
Yeah, can I grab that one?
Yep, sure.
Okay, so the EHR is a good EHR is an invaluable tool for a longitudinal population study, but it is not, and I feel strongly about that, the sole solution for understanding human health or getting answers out of a population cohort. And just a few simple illustrations of that. If you take the disease I've just talked about, diabetes, it's tremendously misclassified based upon the absence of people having an HbA1c test or a glucose test as a matter of routine. So if you follow them up based purely upon EHR, you miss a whole load of people who get diabetes, and if you rely upon the EHR as baseline phenotyping, a lot of your people you thought were healthy actually had diabetes at the outset. Misclassification, which destroys your signatures and the utility of what you find. But then think about, other questions. Precision nutrition. How can we understand nutritional influences unless we assess nutritional intake? The beautiful work that's come out of UK Biobank and imaging, and the DEXA studies, that beautiful paper in Science looking at the genetics of the skeletal system and what it tells us about the evolution of mankind. These things would not be possible from the EHR. And so I passionately believe that if you're going to spend time doing precision molecular phenotyping, 30x depth, long read, or whatever, then you've got to have some precision phenotyping to go alongside it, and it'll bless you in the future.
So I think very briefly, I think this is a major challenge in prospect cohort studies, disease follow-up, disease phenotyping. So at least in Genomic Biobank, we're still trying to improve. Although we are able to link hospital record, we're still trying to do independent outcome adjudication subphenotyping. And also we start to collect sort of discharge notes as well for all the hospital events. So that will be great enhanced. But I agree with John just mentioned. We can also use molecular biomarker to improve, because clinical diagnosis like diabetes is just one disease. But I think with the genetic, proteomic, other, you can actually phenotype them into different kind of molecular subtypes. So this could be quite useful for future clinical practice, for predicting treatment, for the management. So this is actually sort of two-way, not just go to clinical, get more data, but also the research can also inform future kind of diagnosis management as well. So this is a quite important sort of development which we start to see.
I have no additional comment.
No, I'd just like to add something here, just to comment that electronic health records were not designed for science. They were designed for administration and billing and other activities unrelated to science. And although I think we've made the most of them, unless you can collect non-structured data, notes and reports, which is a big challenge, then you're often going to miss some really important phenotypes that are collected in the course of normal healthcare. So I'd like to switch to another question, which I think is at the crux of utilizing the biobanks as a means to really translate. And how do you think about the engagement of the clinical community with your efforts?
I think two ways. One is actually we need to collaborate with the clinicians to ask a good question, because now we've got so much data, we don't know where to start. So that's actually quite important, like chronic renal disease, liver disease, chronic infection. So that's actually quite important. A lot of things can be tested, like proteins mark, got 10,000. So where to start, how to prioritize. So that's quite important things. I mean, another thing is trying to improve disease phenotyping as well. That's quite important clinical input. Third is a translation. I mean, this is something we just start to engage, try to think, drug target, test, and also aging clock, all kind of tools. So it is not good to just publish and then no follow-up. So this is actually, in future, I hope the IHCC could actually try to develop this kind of a dialogue engagement that will be quite useful. Identify some of the, just like for us, top findings has a potential translation. How are we going to push it forward? This could be one result from one study, another, so we can put them together, try to think, plan carefully.
Thank you. Anybody else want to respond to that?
I think it's similar to what I said about international collaboration. Yeah, what we need is clear explanation and start off communication to get a consensus and to develop a shared goal.
Thank you.
I think clinicians should be the heart of a population cohort in a biobank. Their studies designed to help improve human health. And no disrespect for my genetic colleagues or my molecular colleagues. There's a certain perspective that comes with interacting with patients and healthcare that helps you ask the right question and make sure the research you're doing is focused upon why the cohort was set up, which is to improve the health of mankind. So I think clinicians should be at the heart of it. Maybe your question, Jeff, was how do we make sure they are able to be part of it? Because increasingly, data science becomes, and AI, very complicated. So we've got to make sure that we find ways of enabling access in a way that is helps the user and enables them to get to the heart of what they're trying to get to.
Yeah, I would say the heart of this question is really enabling biobanks to address real problems that our clinicians are facing every day and engaging them as the end user of the biobank product. So we're out of time, but I do want to ask each of you to answer in one sentence, what is the next big thing in your biobank? Where are you going from here? What's next? Microphone.
I think it's proteomics, but I really want to also say at the same time, long-read sequencing.
Okay, that's 2 sentences.
Too much.
Zhengming.
I think very similar, whole genome sequence or 500,000, and the proteomics as well. And I can retire.
Okay.
Yeah, multi-omics and more collaboration.
Terrific. There were a lot of really excellent questions that came in, some of which were really specific to the individuals that made their presentations, so I encourage you to come up after we're done here and ask your questions. I really want to thank our speakers for a wonderful session, and all day having a biobank theme was really excellent.